All right, welcome to our third video in the organic molecules lecture. So far we've did an overview, we've discussed carbohydrates, and now we're going to talk about lipids in this video. So lipids, which are also referred to as fats, are a type of organic molecule. And we're going to talk about all of the different properties and the structural components of lipids throughout this video. So carbohydrates are what we refer to as hydrophilic, which means that they are water-loving molecules. So they dissolve easily in water. So think about table sugar, right? So sucrose. You can put table sugar in water, you can stir it, and that sugar will dissolve in water. So you'll get a solution, an aqueous solution when you do that. And so whenever something dissolves in water, it's referred to as being hydrophilic. However, lipids, unlike carbohydrates, are not hydrophilic. They are hydrophobic, so they do not mix well with water. So if you've ever put vegetable oil and poured it in water, you know that if you leave it alone, they will separate and the oil will cause a layer to form above the water. Or below the water, just depending on the density of each solution or each liquid. And lipids are also different from carbohydrates, proteins, and nucleic acids, so the other three organic molecules, in that they are not really made up of repeating monomers. So like carbohydrates, you have repeating glucose molecules that make up starch, and so those monomers make up the polymers. Really, the macromolecules of lipids have kind of a specific structural formula. So for example, a triglycerol has 3 fatty acid tails and a glycerol head. And we'll talk about that in a little bit. But so that what it also means is they're not these really huge, not very large macromolecules in comparison to things like carbohydrates and proteins and nucleic acids. And so lipids are actually made from different molecular building blocks depending on the type of liquid or the type of lipid that we're focusing on. And so we're going to talk about triglycerols, phospholipids, and steroids in this discussion. So here we have an example of oil being hydrophobic and vinegar being hydrophilic, meaning that the oil will not mix with that vinegar solution. And so you have the separation of these two liquids in this container here. And so typically a fat molecule that you probably are thinking about whenever we use the term fat is most often a triglyceride. And a triglyceride molecule is a molecule that's made up of what's known as a glycerol head, a glycerol molecule. And that glycerol molecule will have 3 fatty acid tails that will be linked to the glycerol head via a dehydration reaction. And fats are really important for organisms in that they not only produce energy, but they also produce insulation, and they also help cushion. So for example, fats are used to cushion, say, forces that have hit the body from some external force, and they're used for that method as well as energy and insulation. And so here we have a figure of a triglycerol or a triglyceride, and the triglyceride is a complete triglyceride is this bottom figure here. And this triglyceride is made up of a glycerol head, which is kind of this salmon color, and of 3 fatty acid tails. So you have 1 fatty acid molecule here, and you can see it being joined to that glycerol via dehydration reaction. That happens 2 more times to join 2 more fatty acid tails, and you end up with a glycerol molecule with 3 fatty acid molecules attached to that. And we can look at that process happening in this animation here.
Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long nonpolar hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules. The fatty acid tails in a fat can vary in length. A very important characteristic of fats is the number of double bonds in the hydrocarbon tails. Fats that contain only single bonds are called saturated fats. Fats containing double bonds in 1 or more tails are called unsaturated fats. Unsaturated fats tend to be liquid at room temperature. Vegetable oils are examples. Saturated fats, like butter and lard, are solid at room temperature. Saturated fats in the diet can lead to heart disease, while unsaturated fats are safer.
Okay, so that last part that they were talking about in the video, the saturated versus the unsaturated fats, are really important in affecting the properties of that fat molecule. And so your unsaturated fats is a carbon skeleton that has double bonds in that molecule. And as we saw in the video, those double bonds lead to kind of a bent, kind of kink structure. And because those double bonds kink the structure, it doesn't allow for those fatty acid tails and those triglycerides to really stack tightly together. The kinks kind of make some space in between. And that's why we see unsaturated fats liquid at room temperature. Now, the opposite with saturated fats is that they have single bonds. And so those single bonds means that you're going to have the maximum amount of hydrogen atoms on those carbon atoms as possible. And so, for example, if it's a carbon atom in the middle of the carbon structure, it's going to have 2 hydrogens. If it's on the end, it's going to have 3 hydrogens, etc. And so it's saturated, fully filled with hydrogen atoms, the most it can have. Whereas unsaturated fats, because of that double bond, means they're missing hydrogen atoms that could potentially be there. A saturated fat is much more linear in form, and so it allows it to stack tightly together, whereas again those kinked unsaturated fats, so if this was a kink molecule and it stacks, it's going to have some space like in between my hands there, and that's going to allow it to be liquid at room temperature, which is the opposite for saturated. They're going to be solid at room temperature. And most animal fats are saturated fats. They have a high proportion of saturated fats. And again, they easily stack being solid at room temperature, and that solid fat can build up in your blood vessels and lead to blockage in our arteries, and can lead to atherosclerosis, which can lead to heart disease. Most oils that are found in fishes and plants are unsaturated fats, and those are liquid at room temperature and tend to be much better for your body in terms of dietary fats. There is a process called hydrogenation where we can add hydrogens to unsaturated fats to convert them into saturated fats. And this makes liquid fats solid at room temperature, and it's what creates trans fats. So if you've ever seen trans fats on a dietary label, this is the fat that we're talking about, and it's the type of fat that's typically not good for your body. It can lead to a lot of health problems. So here we have examples of saturated fats. So ice cream, cheese, butter, meat all have a lot of unsaturated fats. Yes, I'm sad. Ice cream I love, but it's saturated and it's not as good for you. Um, we have unsaturated fats, which we see in plant and fish oils, omega-3 fats, which is also in fish and lots of different vegetables and nuts. And then here we have a donut, which is shown to have a trans fat, which is also unfortunate because the things that you love the most are generally the worst for your body. Again, those trans fats are really bad for your body and not typically good to have in your diet. So even though it's a type of unsaturated fat, remember it's converted into a saturated via hydrogenation, which makes it not really good for your body. Steroids are another example of lipids, and steroids have a really different um, structure and function from fats in our body. The structure of steroids is not linear, like we see with, say, the fatty acid tails. They are actually in a ring, so they're made up of 4 carbon rings that have various different functional groups attached to those rings. And the functional groups that are attached to those rings determine what type of steroid it is. And so cholesterol is a type of steroid found in the body, and it's actually the precursor to other steroids such as estrogen and testosterone. So cholesterol will be converted by the body into either estrogen or testosterone, depending on the expression of those in our body, which is usually determined by gender. But cholesterol is a really key component in cellular membranes too. So phospholipid membranes, which we'll talk about when we talk about cells and the structure and function of cells in the next chapter, chapter 4, I believe. So here we have a figure of cholesterol. So you can see the 4 carbon rings that are in yellow, and then the different functional groups that are attached to those 4 carbon rings. And you can notice that the different steroid molecules, so testosterone, estrogen, and cholesterol, have different functional groups attached to those carbon rings for each of the different molecular structures, which again affects the properties and how those steroids are going to work in our body. You can make anabolic steroids, so synthetic anabolic steroids. These are basically variants of testosterone, and they mimic some of the effects of testosterone. Steroids can be used to treat specific diseases. They can be used to treat cancer and AIDS and other types of diseases. But they also can be abused by, say, athletes that are trying to build up muscles quickly to gain an advantage in some sort of way. But again, by taking these steroids, it could lead to problems such as physical and mental problems that could be really bad for the body. And that's why a lot of most athletic organizations ban the use of anabolic steroids for a quote unquote recreational function. All right, so that is it for our lecture video. We didn't talk a lot about phospholipids in this video. Phospholipids are a variation of a lipid molecule, but we're going to talk a lot more about that lipid molecule in the cellular structure and function video. So just keep that in mind. And so yeah, so that's it for our lipid video. The next video in this chapter, we're going to look at the structure and function of proteins. So we'll see you then.